We're at the 22nd CROI. My name is Schaich. I'm with IFARA, and we're here with Sara, Janella, and also Josue, Perez, who are both from UCSD, who are probably from other places earlier, but that's where you're residing at the moment. So what I'm going to ask you, each of you, to do is just introduce yourself a little further, to tell your background and maybe a little bit about how you got into the field.
Okay. Hi, I'm Sara Janella. I am from the Italian part of Switzerland originally, and I am a researcher at the Center for AIDS Research at UCSD. I've been there now since 5 years, and I work in the HIV field. I am very interested in HIV pathogenesis. This means like how HIV gets started and how he behaves in the body during the earliest phase of HIV infection. And I'm also very interested in how HIV behaves in anatomic compartment, like for example the genital tract and the central nervous system. And yeah, this is what we will talk about today, right?
That's right.
So I am originally from Puerto Rico. I came to the US in 2006. I started doing my PhD at UCSD. I did my degree in bioinformatics, and actually I've been working in the same lab for a while now. As a student, I worked with understanding the influence of the gut flora during HIV infection and during antiretroviral therapy. And more recently, I've been interested in understanding the neuropathogenesis of HIVs, like factors that can contribute to persistent inflammation within the central nervous system, which is what brought me actually to CROI. I have a poster about that. And even more recent, I'm trying to join the two topics, how the gut really influences like outcome that has to do with neuroaids. So I'm trying to connect those two right now.
That seems really interesting. I mean, it seems to me like it should be a connection, and theoretically. But how do you propose to understand that? Is there a logical pathway that would allow you to follow it?
There's been few studies done in mice outside the HIV field that they have done what is called fecal transplants, and they see how mice actually change behavior. Some mice that they were calm and got the fecal particles from a mice that is anxious, then the calm mouse end up being anxious, and something also shyness versus social mice. So what we plan to do, and Sara here is actually helping with this project, we're gonna try to collect like pair sample, like stool samples and like cerebrospinal fluid from the same people, and then look at the gut composition and all these other markers of inflammation, systemic and in the central nervous system, and try to then fish out the pathways, how what we see in the gut is really associated to factors like phenomenon like microbial translocation, like how having certain flora may or may not like aggravate this leaky gut condition that ends up contributing to activation of the immune system, like systemically in the blood, and how that even makes the blood-brain barrier more permeable, so that all these bacterial products that cause inflammation now also go to the central nervous system, causing then inflammation. We don't know if this is like a compartmentalized problem, or it's just throughout. So I don't think, and Sara might correct me, I don't think in the HIV field people actually have been looking at this yet. So we're trying to go to that route.
So is inflammation causal or a result? I think that's the question, because
I would say both. I would say both. I don't think it's black and white. I mean, of course there are components that comes from the virus, right? And all related to virus. Like you have all these CD4 depletions because of the virus, so that already makes an immune regulation. So, and you have the immune system trying to attack the virus. But then you have all this collateral damage, right? Like all this depletion in the gut, for example, makes the gut permeable. And then now you're having all these bacterial products that normally in the gut, they don't cause any problem because the gut is used to this. But once they pass to the blood, then the body says, this should not be here. So it starts attacking that. And now you have inflammation that is not really directly related to the virus, but it's just what we call like collateral damage, right? Now all these old products are there, the system is going to try to attack it, and then you get inflammation from there too. And then as that we have other examples of indirect causes of inflammation.
It seems like the immune system is so redundant. It has the innate and the adaptive. It has, and like you say, and I think that, but it seems as though when the virus is first, when person zero converts, they have this natural progression. The first thing seems to be is the gut is the worst affected.
Yes.
So that would be the primary thing that you might evaluate. But it seems like as soon as a person's infected, it seems like their body architecture and the way the whole body works is basically reconfigured. Is that a fair statement?
I mean, it's complicated.
Yeah. The body's trying to fight to reestablish the norm, but the norm is now a new norm.
Yeah, exactly. I mean, even with successful therapy, the body is never close to what it was before infection, and this is the main problem of having HIV. I mean, antiretrovirals are very successful these days. Like, 20 years ago, people had to take like 25 pills a day. Now, probably, I think, I'm not a clinician, but I think they only take one, right, a day, and all the combination is all in one pill. But systemically, people never go back to what they were before infection, at least in terms of inflammation and immune activation, and that's the main problem with progressing to AIDS. This constant immune activation that the body experiences is basically the driver to AIDS, and no one really knows how to try to put it back to where it was. It goes with therapy, yes, it reduces, but it never goes back to where it was before.
I think that one of the most important things is that also the best shot that we have to limit the damage is to just start antiretroviral therapy as early as possible during infection. And this brings to the point that in reality it's very important to just diagnose HIV as early as possible, to have people tested, to have people be aware of their infection, have access to antiretroviral therapy early.
And they can have less of a course, a better course of disease.
Absolutely.
Yeah.
Also, I think that early antiretroviral therapy is the one thing that nobody will argue about that can give you a better prognosis and like a better disease outcome.
And we were talking before the program began about that there are drugs that seem to be more, we used to be very concerned about that, whether the drugs cross the blood-brain barrier, because that is your central nervous system and that's what you're working on. But what is the challenge there? Do you feel that it's really important to make sure that those drugs are drugs that cross the blood-brain barrier?
Yeah, absolutely.
Yeah. Or at least maybe some. I know that when you're taking a combo, it may be that some of them do and some of them don't. Does that make any difference, do you think? Or do we not know that? Maybe we...
No, we do know that. So there is plenty of research in the past and in the present to kind of understand how important it is to have drugs that cross the blood-brain barrier. And I think that now we know it is important, and actually the new drugs more and more are taking care of that. They actually really penetrate the tissue and the compartment and the blood-brain barrier in a way that they can effectively target the virus in every compartment. I think that's where we strike, right? This is what we want from our drug, that it penetrates.
I want to kind of draw a little bit of a temporary conclusion here, that we are suggesting that when you become positive, you have certain changes in your body that will never go back to what they were before. And these are somewhat consequential, maybe minimalized by antiretroviral treatment. So it makes sense for people not to become positive. Today, people do not seem to have that sense of urgency or importance centered around staying negative. I don't know where that's coming from, but maybe they're just fatigue of trying to avoid HIV, I don't know. But we have demonstrated, you mentioned, you confirmed, this is something that we don't want to have happen because it is not a good thing to become positive. Having said that, if the person does become positive, we need to know it right away because there is a better course of disease that they will take. The pathogenesis will be a little bit improved by the sense that they will be early treated. Because, and the other thing is, I think that people don't understand is that there is many times a way to identify onset of HIV by the flu symptomology. So if you have otherwise not well understood why you have flu symptoms or a horrible flu-like condition, this could very well be onset of HIV. The matter of days before, I think, or something like that, I think that's what we're talking about. Within a matter of days you were infected, you will come down with a huge flu because your virus goes nuts. It goes way up and then it comes down abruptly, so you get over the flu, but basically your body is now being seeded with the virus. At that point, you can address by taking antivirals and getting a better course of disease. Certainly then, if not even a little bit later. So that's just a point that I wanted to bring up because I like to draw some extrapolations to the importance of certain very natural things that people need to do.
And
No, you go.
okay, no, I just wanted to add that, of course, you want to avoid getting infected with HIV,
yeah.
But if, unfortunately, you are one of this person that gets infected, people have to understand that we have to be responsible about it. Because I have heard, from people from the community, like, oh, I am infected, so I don't care. I will have all this, behaviors, like people think that, for example, they cannot get super infected. You can get infected more than once, and that will even complicate even more, the outcome of HIV, because now you have two different viruses in your system, so everything is accelerated. It's very common also that people get...
So the superinfection would happen because a person was not treated.
Well, it can happen because a lot of things. If people are careless, they don't go to treatment, and they're having all this
casual
behavioral risk, and they're not responsible about it. Then they're exposed, and they're exposing other people, right? So if they're with someone or with that is also careless, and they have some sort of sexual intercourse, one person can transmit the virus to the other person, even though they're infected already. And then everything is accelerated because now you have two viruses in your system
with different resistances and so forth.
They're totally different.
It's called serosorting. Actually, we know that people do that, right? People that So
people think that this cannot happen, and this is why I said we have to be responsible. We have to educate ourselves because having HIV at this point in life is not the end of the world anymore, like in the 80s, So, people, if they are responsible enough and they go to care, they can live forever, But
they still have to be responsible.
But it's a decision that people have to come up with. It's like, I want to be better at this. I will go to care, I will be honest with my sexual partners, I will, be communicative. And as long as you educate yourself and know what you're doing and that you're doing it right, that, sure, HIV is a problem, but it's not the end.
So, would you say, now we were talking about the blood-brain barrier. Can you give a little, this is more or less your expertise, so can you just give a little more expansion on that? Because I don't think people may understand what this is. I mean, our audience is diverse.
Okay.
So.
Yeah, actually, there's what we call the blood-brain barrier. Is just like a membrane that that separates everything that is in the central nervous system, that is the blood, the cerebrospinal fluid, the spinal cord, and the brain, from what is outside. And as you can imagine, it needs to regulate the traffic of cells and protein, because we don't want everything to go into the brain, but we want something to go into the brain. And so having the right level of permeability, so the body, of course, is very good in what he does, right? He can regulate exactly what goes into the brain in a normal situation, but of course, when inflammation occurs, this blood-brain barrier can become more permeable, and so we have more toxic product and cells trafficking into the brain, and this can damage the brain, can cause neurocognitive impairment, and this is part of what happened in HIV and also in all other kind of disease that affect the brain. So in HIV specifically, I think that one of the important questions that we're trying to answer right now is if HIV is able to maintain a viral reservoir. So as, I don't know, probably people know, HIV has this characteristic that he integrates into the human genome, and once it's integrated, our antiretroviral therapy cannot affect it anymore. So once it's integrated, it's there, and we have no way right now with the current therapy to eliminate it. And this is the main reason why people cannot be cured from HIV right now. And
it's one of the compartments. We talk about compartments. It's not like over here or over there. It's a body system. One being the brain, blood-brain barrier, etc.
So when we talk about compartment, usually in the field, we have the blood and the lymph tissue. That is what everybody, like the more obvious place to look for HIV. And then we have the called anatomic compartment, that is the brain, the genital tract, the gut. That these are like a location that are a little bit more difficult and tricky to reach, and that we worry that maybe some residual HIV can just sit in there, and that we
without waking up, or and that's part of what we talk about when we do programs on the cure. We talk about that, activating the virus so that we can attack it and make it do battles.
But this is one of the questions. So if we do this activating the virus and attack it, are we really doing it everywhere, or maybe we cannot reach every single corner of our body? Like, for example, the brain. The brain is one of those, because of the blood-brain barrier that kind of prevents cell and stuff to go into the brain like unrestricted. There is always a little bit of a worry that maybe we succeed in clearing the viral reservoir all over the body, and then you still have one cell in the brain that just...
And that's all it takes.
And that's all it takes.
I'll be talking with, I don't know if you know Tim Henrich, who did this study on the two gentlemen up in Boston, the Boston patients.
Oh, yes, yeah.
And I will be doing a follow-up with him, and I'm sure that's what we're going to be talking about, how this little bit of virus, wherever it was, after so long came out. And it just, and we don't know whether it's a number of two, so we really can't get good results from that. But it's hard to find, to do a study with 100, 200 people. But that's probably what you need to know. Is there any possibility of finding anybody where we can get every compartment and every morsel or every particle of HIV, which is what is necessary to perform some sort of sterilizing cure.
Yeah. So this happened once, right?
Yeah, it happened once, but that's total ablation. It's like it's like a 50-50, maybe even worse than 50-50 chance.
Yeah, no, that's
and he had to do it anyway. So, but taking a healthy person otherwise that would just manage to be on treatment the rest of their lives and not go through that risk. So and also very expensive. we talked about that in the cure workshop. Horribly expensive, millions of dollars. It's not realistic. So we have to think in terms of what we can do to manage the virus and put it in the best possible livable state. I guess that's the best way to put it in simple terms. So we can live with the virus. And we talked about that day one, when we did it before we even had drugs. Mentally living with the virus, so you didn't feel like you were, obviously, I think anyone that becomes positive today will go through that moment where it's very different when you know you have the virus and you're living with it, as opposed to when you're on the other side of the fence and you know you don't have it. So we're looking at it's a psychological moment, I think, that transforms you in many other ways. So this program kind of covered a little bit about prevention mentally, and also a little bit about treatment and about understanding the science. And I really appreciate you all for making this so possible, because you're energized. We talked about having Scott here, your colleague, who's extremely good at all of this. He's a broad sense of knowledge, but couldn't be here with us today, and you did fabulously.
Well, we are Scott's kids, right?
Yeah, we're Scott kids.
Exactly. There you go. There you go. Thank you so much for being here.
Thank you for having us.
Thank you.
Thank you.